It happens a lot. Okay, so we're going to continue here just to wrap up on some important concepts that have to do with that complement thing. And then we're going to go to look at antibodies. Well, welcome, just in time. All right, so finishing up the idea of how the complement is working and important receptors that are working with those floating complement proteins and their importance on activating certain aspects of the system in general. Okay, so we had mentioned before that we have those receptors on surface of cells that can interact with cleaved complement stuff, right? So we mentioned here that we have complement receptor 1, right? That thing is the same as CD35, so it's the same name for this receptor on the cell surface, okay? So what is this receptor? This thing is a glycoprotein, so it's a protein that has some sugars stuck to it, and it is bound to the membrane. Okay, so it's membrane bound, and this receptor, let's put a picture here. So here we have a cell, that's the nucleus, and here we have this complement. This is complement CR1, the complement receptor 1, bound on the surface of the cell. It has some amino acids, it has some sugars, so that's what you call a glycoprotein, right? And what is it going to do? It is going to bind to cleaved C3b and also C4b. Where are you going to find C3b and C4b? Where do you have it? Okay, what is C3b? It's a simple question. Okay, I got incentives. Chocolate. No, C3b. How do you get non-convertase? No. Okay, I hope I have enough chocolate. Okay, so you said something. Yes, you're. All right. On the surface of the pathogen. So here we get, everybody heard that, right? So here we have, let's draw a pathogen here, green pathogen. Let's say this thing is a bacterium, right? So we do not put nucleus. It just has a DNA, circular DNA. So here, circular DNA right there. Okay, right there. That's bacterium. It so happened that you had those complement proteins always floating around, like C3, right? But then remember C3, it can do the autocleaving, and it can become C3b that binds to the surface of the pathogen, right? C3b. And then you have C3a, right? The other piece here, that is C3a, that is floating away, right? So C3b on the surface of the pathogen is tagging the pathogen. So C3b is the opsonin, right? So it did opsonization. It's tagging the pathogen right there. So this guy here, CR1, hello, CR1 on the surface of some cells, right? CR1 is going to bind to C3b right there. Okay, so let's see what is happening here. CR1 on the surface of some cells is going to bind to C3b, also C4b, but let's think C3b on the surface of pathogen. Okay, now what is going to happen here? First, you have to think, where is it that you're going to find CR1? Okay, then you're going to think what's going to happen. CR1, where is CR1? You're going to find it on the surface of white blood cells, so those are your leukocytes, okay? And you're also going to find on the surface of your red blood cells, that's your erythrocytes, right? Everybody cool with that? So this guy over here is a white blood cell, right? It has CR1 on the surface. This guy is going to bind to the opsonized pathogens. So now let's see what's happening here. On those white blood cells that are part of the innate immune system, this you're going to find abundance of CR1, especially on cells that can eat up, that can do the phagocytosis. Okay, so on the phagocytosis, it's going to bind the tagged bacterium, and what's it going to do? It's going to eat it. It's going to say, I could eat the bacterium just by itself, but now it got seasoning. It is tastier. Okay, so this is basically how it acts. It goes like, ooh, I like that seasoning. I'm going to eat it better. Okay, and it eats it better. Not only it likes it, to eat it better, it's also going to receive information from this interaction to upregulate those oxidative pathways, the what do you call them? Oxidative burst or respiratory burst. Remember the guy? Remember how he killed phagocytosed the pathogens, right? You have 3 stages of killing the pathogen. First one is just by regular enzymes, right? The other one is by acidified environment that you get the azurophilic enzyme. And then the third and the most effective way is by oxidative respiration, right? By oxidative burst, okay? Oxidative burst. So this is helping in several areas. Is helping to phagocytose, is activating the killing of the phagocytosed pathogen, all right? When you have here on your phagocytes, okay? Now, besides being on the surface of those innate cells, those phagocytes, it also is present on the surface of B cells, okay? So what is the B cell going to do? The B cell is also going to interact with a tagged pathogen and is also going to phagocytose it. Okay, B cells also do phagocytosis. They are not known as phagocytes because that's not their most important job, but they can internalize pathogens and they can chop up pathogens, okay? And they can even present a piece of the pathogen to T cell, okay? But as we're going to see later, B cells are not very good at doing this job, okay? They are good at presenting pathogens to T cells that are not naive. They can present pathogen to T cell that is already experienced, because that goes in details later. So B cells can also phagocytose. They can process pathogen, and they can help to activate effector T cells. Okay, so B cells have CR1 on their surface. Okay, so these guys here are using CR1 for phagocytosis. Okay, then you're going to find CR1 on the surface of those red blood cells, and this is the part that is so interesting that puts red blood cells as an effector cell for immunity, okay? So we thought red blood cells only had the job of transporting oxygen and some carbon dioxide, right? But no, the red blood cells also have an important role in immune system, okay? With this, let's do it here, the red blood cell. Okay, so here is your red cell with your CR1 on the surface of your red blood cell. This thing is capable of binding, look at this, binding complexes of antibodies and antigens, okay, including binding here to C3b, okay, that is bound to antigen, okay, binding these complexes and bringing those to the liver or to the spleen for destruction of whatever it brings there. Okay, antibody-antigen complex, and you can also bind this C3b bound to pathogen. Okay, you got the point, you got the job of red blood cell here in immunity. Okay, so it can catch some tagged or some opsonized pathogens and help to destroy them. Okay. Okay. To the antigens, got it. Okay, so a lot of information on that slide. All right. So here, what happens if you have a problem with that receptor, with this CR1 receptor? What kind of diseases are most common with when you have problems with CR1? Okay, look at this. Variants in the gene that makes CR1, by the way, CR1 is on chromosome 1, the biggest chromosome, right? CR1, problems with the gene is involved in Alzheimer's disease, but not early onset, is late Alzheimer's disease, okay? And is also involved in how susceptible you are to certain infections, including viral infections, okay? You can imagine that if you have a problem of phagocytosing opsonized pathogens, the pathogen can get away from getting killed, and then you can have problem of recurring infections. Right, in general, every type of bacterial infection can reoccur here. Okay, so problem with CR1 is a big problem. Okay, now not only you have susceptibility to bacterial infection, but you also get more susceptible to viral types of infections, including here CD19, right? I mean COVID-19, right? As we had seen before during the pandemic. We have seen certain people that were more susceptible to developing COVID in comparison to other people that were exposed but didn't quite develop severe COVID symptoms, right, or signs. Okay, so that could also do with variation of CR1 gene expression. Okay, all right. Now another thing is that even if you have a proper gene, but let's say now that instead of having proper levels of protein expression, let's say that you have lower level of protein expression, right? That can make a person also susceptible to developing lupus. You heard of lupus? Yeah, it's a type of autoimmune disease, right? So susceptibility to developing lupus. Right? So that's your clinical correlation. Okay. And then you have this other receptor, this other guy that is CR2, right? So you have complement receptor 1, and now you have complement receptor 2. CR2 is also known as CD21. Yeah, I know those CDs are just big time nightmares, right? So just think complement receptor 2, okay? So this guy is on the surface of cells, but this guy here is a transmembrane, okay? Transmembrane glycoprotein. Yeah, transmembrane glycoprotein. And you're going to have some specific cells that have them, that has this CD20, okay? So let's see what is happening here. What are the type of cells that you're going to have? Importantly, you're going to find this on the surface of mature B cells, okay? Also follicular dendritic cells. Those are the guys that are going to help the B cells to mature, okay? Those are not just any type of B cell that is floating around. Follicular dendritic cells are helping the B cells to mature, okay? And you have some on the surface of T cells, okay? So look at this thing here. This CD21 is not just only on the surface, it's a transmembrane, okay? Because the inside part is is going to send signal transduction. By the way, CR1 is also a transmembrane. Okay, so this guy, transmembrane glycoprotein. Okay, here on the surface of these cells, this receptor is going to bind to a piece of C3 that got cleaved. But this C3 here is not C3b, it's another piece, it's called C3d. Okay, but it's a piece of C3 that got cleaved. When you cut C3, you get C3a, C3b, you also get C3d, it's a variation of the C3b that got cut. Okay, what is going to happen here? C3d also binds to the pathogen, is a variation of C3b. Okay, so we could call by all practical terms, we could call it C3b. Okay, so is C3 bound to the antigen? Okay, this receptor is going to bind to a piece of the complement that is bound to antigen. Okay, so what is happening here? The antigen is tagged, is opsonized, it interacts with the receptor, and this receptor is on the surface of a B cell. The receptor is on the surface of the B cell. Why is it important for this interaction to take place? Well, we're going to see when we find activation of B cells, we're going to see that on the surface of B cells, you have the B cell receptor, which is also known as an antibody, right? I'm going to see today, this is a type of antibody that's called IgM on the surface of the B cell. Okay, so this B cell receptor has a piece that recognizes directly the antigen, right? So the surface here, the very tip of this antibody has some binding sites, two binding sites that directly bind to the antigen, to the whole antigen. Okay, so this is important to keep in mind how the B cell recognizes antigen, right? It recognizes by 3D, it binds the whole shape of the antigen, like you can bind two of them, okay? It grabs and it touches on 3D and is binding and it feels what's the antigen. It's very specific for an antigen, okay? So what's going to happen here? If you have only the B cell receptor binding to antigen, then you're not going to really get a response, or you're not going to get the B cell to do things that it's supposed to do. Okay, so only one binding of antigen is not enough to activate your B cell. Now, if your antigen comes tagged, and the tag interacts with this other receptor, that is your CR2, now you can activate your B cell action. Okay, because now you're going to have one signal that comes down the B cell receptor and another signal that is going to come down CR2. And in order to activate the B cells, to make them effector cells, you need to have at least two signals. Okay, so this receptor here is very important to send out this second signal for B cell activation. Okay, so what can it bind? It can bind ICB3, it can bind this other C3d type of fragment. Okay, and the importance of this receptor is that it is really connecting the innate immune system, because this thing is just the same in all of the cells, right? You're not modifying it, it's not part of adaptive system. It is interacting with the innate immune components and bridging it with adaptive, because antibodies are part of adaptive immune system. Okay? So antibody functioning on the surface of B cell is already part of your adaptive immune system. So this receptor is really bridging the innate and the adaptive type of response. Okay, and is important for activating your B cells. Okay. So this is what it means, but when I say here that provides a secondary signal to activate the B cell. Okay, effective activation of your B cell. Okay, everybody got this? All right. Okay. All right, remember your C3b is that opsonin, all right, that's what coats the vesicle, I mean the pathogen, right, it coats the pathogen. And here, just to reiterate that you have your white blood cells and your red blood cells, they have CR1, okay, and this is just a little review slide, okay. Second, what happened here? Okay. Right in the middle. Okay. Okay, so what is saying here? The little pieces that fly away, remember those guys here, C3a and C5a, like right in the beginning before you get the MAC complex formation. So 3a and C3a and C5a, they have very, very important jobs when they fly away from the cleavage site, right? They are both chemoattractants, right? So they are known to attract white blood cells to this site of inflammation. Okay, what kind of white blood cells? They're going to bring the phagocytes. So you have your neutrophils and macrophages. Okay, they're going to bring eosinophils and monocytes, right? And remember also, I forgot to mention that these guys here, the neutrophils, when they come to the site of inflammation, they are not only very good phagocytic cells, right? They are one of the most potent phagocytic cells, but they have a very neat way to trap pathogens. Okay, they go through this process that is called NET, neutrophil. I forgot what the ET stands for. Okay, I remember this because this neutrophil, first, what it's going to do is going to sacrifice itself. Okay, if it sees a lot of pathogens floating around, a lot of bacteria, it says, well, I can't eat it all, but I can trap them in a place. And you see how the nucleus has all these lobes, right? They are known for this polymorphonuclear type of nucleus, right? So what the neutrophil does, it gets the chromosomes, its own chromosomes, and it uses it as a big fishing net. Have you seen these people that go fishing and they have this net and they throw it like that in the ocean and then they grab a lot of fish? You know what I'm talking about? Like if you watch those old movies and they have like, you know, those religious movies and they have the fishermen that they throw the fish, they throw the net, right, catch the fish. That's what I'm talking about. So neutrophils like to do that too. They use their own DNA, the strings of DNA, as a net. They throw the DNA on top of the pathogens and trap them there. And then they wait for other phagocytic cells to get there and eat the fish, I mean the bacteria. But that's a very, very neat way that these guys have to really try to avoid dispersion and systemic invasion of the pathogens that are at the site of inflammation. Okay. The presence of these guys over here help with this process of activating those leukocytes. Okay, so these guys are not only chemotaxins, but they also are anaphylatoxins. They're anaphylatoxins. Okay, that means they activate the effector function of those white blood cells, including the NET formation. Okay, and they are going to activate those granulocytes to do the degranulation. They bind to receptors on those cells and allow for these granules full of toxic stuff to be dumped at the site of inflammation. Okay, so this is what they're doing here. They are pro-inflammatory, and they are both doing this job. C5a is even more potent than C3a. Okay, but they have this redundant type of function. Okay, so this is what is happening with your granulocytes, okay, and also with other white blood cells that have granules that you don't see them, with agranulocytes as well, okay? So what is happening here? You have here your C3a and you see C5a. They bind to a receptor, and look at this thing here. This is a transmembrane, and it happened to be G protein couple receptor. So you look at this, it has 1, 2, 3, 4, 5, 6, 7 transmembrane protein with a binding extracellular domain and active intracellular domain, right? So this G protein couple receptor, after it binds to this signal, it is going to send transmembrane signal. It is going to send a message to the inside of the cell for degranulation. Okay, so depending on the cell type, they all have this receptor, right? What's going to happen? You're going to have activation of phagocytosis. You're going to still have cytokine production from these cells. Okay, here degranulation, right? This is a big one. Degranulation. Okay. Oxidative burst activation. Okay. So you got the point, right? With these things around, it's going to bind to G protein couple receptor, and you're going to activate the cells to do the things they're supposed to do at the inflammatory site. So here's a picture showing that. You have here your C5a that has here the receptor on your mast cell. Okay, this guy's a mast cell. You see C5a plugs in to the receptor, and then you have these effects going on. So what is happening here? You have histamine that is dumped. What is histamine going to do? Inflammation. Inflammation like what? How? Vasodilation and vasodilation, and what else happened to the blood vessels? Increased vascular permeability, right? Vasodilation, vascular permeability. So histamine releasing, here same thing here. Here you have your C5a, here you have your C3a binding to that receptor. Okay, so that's what is happening here. Okay, showing those cells have the receptors too. All right. All right, so overall picture of what's happening here. You have your lower number of complement systems that are very much important for chemotaxis, anaphylatoxin, right, and tagging of antigen, right, and they're going to be important for formation of complexes, right. So we can see that if you have any type of deficiency in your lower complement proteins, you're going to have more severe health problems, okay, in comparison than if you have deficiencies with the higher number of complement proteins, okay? So if you have a problem with either C1 or C2 or C3 or C4, you're going to have problems like lupus. You're going to have a huge problem with repeated infections, right? And I did mention that before, and there was even a test question that was implying what's happening here with the complement, right? So you're going to have problems of autoimmune diseases, also some kidney disease with repeated infection. Generalized type of infections is a big problem. Okay. Now remember, if you have a problem with C3, that's the biggest problem you can have. Okay, because C3 is really in the middle of the three pathways. All right. Now, Lower number, bigger problem. If you have a problem with expressing the higher numbers of the complement proteins, like C5, 6, 7, or 6, 7, or 8, okay? Now you end up developing one specific type of recurrent infection. And this has to do with this specific type of gram-negative bacteria in specific Neisseria type of bacteria. So you basically have two Neisserias that cause disease, right, in humans. You have one that is Neisseria meningitis, and the other one that is Neisseria gonorrhea, right? What can Neisseria meningitis cause? Meningitis. And Neisseria gonorrhea? The gonorrhea, right? But it can also cause meningitis, huh? But then the meningitis usually does not cause the gonorrhea. Okay, go figure that one. Okay, so here, what is happening here? If you have this gram-negative bacteria, usually that gram-negative bacteria has this outer membrane, right? And it also has a capsule, is encapsulated, right? So what happens here is that the phagocytes get in there, and usually they don't eat this bacterium very easily. So they'll need to get opsonized, guys. They need to get opsonized and to be phagocytosed. So let's say here they are opsonized and phagocytosed. But then once you have your C3 and C4 complex here, then you want to get your C5 convertase. Remember, what is your C5 convertase? What is the C5 convertase? You have two of them. Okay, for C5 convertase, you have C3B, B, this guy is a C3 convertase, right? C3 convertase. But then, when you get more C3B on the surface, you can get another C3B that gets here. And this here, yeah, watch the name, right? C3B, BB, C3B. That is your C5 convertase. Okay, and which pathway is that gives you this C5 convertase? Okay, it's not the classical, so you have another. Alternative. Okay, you get it, John. Hopefully you're not allergic to nuts. Ready? Oh, good catch. All right. Alternative pathway. This is your C3 convertase of the alternative pathway, C3BBB, okay? Then when you get C3BBB, C3B, that is your C5 convertase for your alternative pathway, okay? Now, what is your C5 convertase for either mannose binding or either lectin or classical pathway? Well, let's see. First, you have to see what is your C3 convertase for those. What is that? Is C4B, C2A. This is your C3 convertase, right? C3 convertase. Now, when you get C3B on the surface, just you get C3B here. Now this whole thing becomes a C5 convertase. Okay, so C5 convertase of the other two pathways is C4B, C2A, C3B. Okay, it's easier just to think of the little pieces binding on the surface instead of their names, okay? Now what's going to happen? C5 convertase is going to get C5 and break it into C5B and C5A. C5B goes on the surface and collects the other guys that do not need to be cut. So you get C5, you get C6, C7, C8. That is enough to make a pore, to make a hole on your bacteria. Okay, so these guys here are enough to make holes and burst the bacteria. If you have a deficiency in one of those, you're going to have a harder time punching holes on the surface of that type of bacteria. Okay? That means you're going to have recurrent Neisseria infections. Okay? Recurrent Neisseria type of infections, including recurrent meningitis, life-threatening. Okay? So you get the picture here. So problem with the higher numbers give you recurrent Neisseria infections. Problem with the lower numbers give you a lot more problems. A lot more problems of all generalized type of infections and autoimmune diseases as well. And kidney failure, kidney problems. Okay? Okay, so these guys over here. Now, if you have a deficiency in this last one here, C9, then you don't have problems. Okay? Because C9 is just an added punch or added hole to the bacterium, but if you don't have C9, the other guys here already take care of the problem of bursting the membrane. Yeah.
I have a question. For the first one, just comment to my mind. Is there any way to know how much like efficiency in C5 or like any other numbers, and those antibiotic affects that or no?
That's a great question. So the question is, can you avoid these problems? Can you correct the problems? Now, I want you to think about it, the whole class, think about it. What are these things? What are they? They're proteins. They're proteins. Where are they found? Where do you find the proteins? The genes are coding for the proteins, but where do you find the proteins themselves? In the blood, in the lymph. So if you are having recurrent issue with Neisseria infection, you run a blood test and you look for levels of those proteins, and you find, for example, that C3 is missing or is very, very low. What can you do? First, you always think about giving antibiotics because you got an active infection going on. But besides antibiotics, what else can you do? Well, you're thinking too much. It would be nice if the liver would produce more if you only have deficiency in the production. No, if you have the zymogen form of the protein. What else can you do? You treat the patient with the protein. Yeah, you were going to say that. Okay, you got to check. I read your mind. You don't want to chocolate? Okay. Oh, sorry. It's covered. Okay. You can infuse, you can just put it in the blood. You put the protein, okay? You have recombinant proteins, right? You produce them in the lab, just like you have insulin, right? And you treat your patient. That's a very effective drug treatment. Of course, you cannot put too much of it, because if it starts to autoclave like crazy, then you're going to have other problems. But you have to have a certain basal level of those working proteins to normal levels, right? So it's very, very important to think, okay, if my patient has this issue, how can I help the patient? Right? Oh, he's missing this protein. Okay, that's easy. We're not going to go with the gene therapy. That would be even better, right? Just substitute the gene that is with the defect, and now that's it. The patient is cured. But if you don't have that readily available, you can just put the protein that is missing. Okay? Yeah, IV. Yeah, is the injection. Yeah.
Once you are, let's say, like stay there permanently or can go down?
Well, that's the thing. You should have normal levels until you're free from infection, until your body gets rid of the pathogen. But I don't know, I'm not a clinician, so I don't know how the continuous treatment of those patients, how it goes. Maybe they keep on, monitoring for the levels, because remember, those proteins do not hang around for a long time. They have spatial and temporal type of control. So maybe they have more stabilized forms of those proteins for treatment. So I'm not really sure how the treatment goes. Okay, but you think about it. If you're defecting on a protein, it makes sense to give the protein. Okay. All right, so here is another little connection if you're playing that video game, right, on the Immune Quest. So this stage of the video game, after you break the physical barrier, this is when you look at host functions to protect yourself from those complement proteins, right? Remember the key accelerating factor, okay, the DAF duct, and protectin, right? And remember also you have the factor I to inactivate C3b. So this is all part of the Immune Quest part. All right. And here I have 3 slides just to summarize the 3 different pathways. I find they get in handy. And they put them together. And here is just for you to have some extra practice on what is happening with the systems. Okay. Okay, finally, we get here to antigens and antibodies, and this is a relatively simple concept. Okay, so these are all of the aims. Okay, so here, first thing I'm going to do here is to think what are the cells that are involved in what you call humoral immunity. Okay, humoral immunity is the immunity with the humors, with the fluids. The first thing you're going to think about is antibodies. Okay? Although complement proteins are also part of your fluids immunity, but when you think about humoral immunity, you're also going to include the fluids that are part of your mucous membranes. So in that sense, you're really talking about antibodies. Okay, so humoral immunity, eating antibodies, and what are the cells that produce antibodies? Those are the plasma cells that are the mature B cells. Okay, so B cells are the only ones that are going to produce antibodies. Okay, B cell activity. All right, we're going to look here at the structure of antibody and what their functions are. All right, so here, this is a lot of information in this slide. Okay, I mean, a lot. It's a whole summary of all of the functions of the antibodies here. Okay, what are they going to do? They work as mediators to get rid of pathogens, but they do not do the killing by themselves. Okay, antibodies cannot kill, but they can mediate the killing, right? They can tag, they can mediate the killing. So what is it that they are doing? Well, depending on what kind of beast it found, it's going to do different things. This is why you have different classes of antibodies. They have to adapt the attack depending on how the pathogen has to die. Okay? So what is the attack? Attack is what you call the effector functions of the antibodies. All right, so let's see here. The first type of attack that they can do is attack by neutralization. Okay, so what does that mean? That means that when the antibody binds to the surface of the pathogen, it is going to block the interaction of that pathogen to its target cell. Okay, so pathogen was nicely happy looking for the target, looking where it should interact and infect, and now it got blocked. It has this big thing that is not allowing it to interact with something else. So this is the first function, neutralization. Okay? Not only antibodies can neutralize the pathogen, they can also neutralize toxins that the pathogens are producing. Okay? Because sometimes, even if you kill the pathogen, if you have the toxins that are left behind in the system, the toxins can do more harm than the pathogens themselves. Okay? So antibodies are important to neutralizing the toxins and not allowing the toxin to reach their target cell to damage the target cell, right? Neutralization, that's the first thing. Second thing the antibodies can do is agglutination, right? That is a related property related to neutralization, because when you talk neutralization, you are talking about neutralizing one bacterium right there. When you're talking about agglutination, now your bacterium is, let's say this guy here is neutralized, but now you're gonna get a lot of those guys stuck together in a clump, so that this whole clump can be dealt with, and this whole thing can go and be destroyed together, okay? So agglutination is just clumping stuff together. Of what? Of your antibodies and your antigens. And the property that allows for agglutination is that each antibody is going to have 2 binding sites, at least 2 binding sites. Okay, so here you see that this antibody here bound to one bacterium, but then another antibody and bound to the same bacterium, and this guy can bind to 2 bacteria. And this guy is also bound to this other antibody, and this one can bind to another antibody, and you get this, you know, let's hold hands and get everything in a clump. So that is agglutination. Okay, everybody clear with the agglutination? All right. Third property of antibodies is tagging, so opsonization. So this property we have seen before with C3b and C4b, the opsonization part. Okay, so what you're going to do, you tag by binding to your pathogen, and you can say, oh, isn't that neutralization? Oh yeah, but you don't want just to neutralize it, you want other cells to go there and eat the pathogen. So the one piece of the antibody that is called your constant region, your piece of the antibody can interact with the receptors of phagocytes and do the same type of work that your C3b did, okay, with CR1. So now what's going to happen here? You have antibody receptor that bind to the antibody that is connected to the pathogen, and you activate your phagocytic cell to eat up the pathogen. Right. So this is your third effector function, opsonization. Right. Right. Now let's look here at the fourth effector function that we started to look at it before, which is complement activation. Which complement? Classical pathway. Here is your C1 cucumber. The big one is a cucumber, right? C1QRS. And if you get those antibodies that activate the conformation of this C1 complex, you are going to have complement activation, and you're going to have the MAC punching holes on the pathogen. What are the 2 antibodies that are good at activating complement? Two types. What are the 2 types of antibodies that are good at activating complement? IgG and IgM, right? IgG. No, this is not working. No, I have battery. IgG and IgM. Those are good for complement activation, right? Okay, and then you have another function of antibody here, and this is a cool one. Antibody-dependent cell-mediated cytotoxicity, or ADCC. It's not ACDC, though you're too young for that. You know what I'm talking about? The rock band. I don't know what they sing, but it's very loud. Okay, no, ADCC. Okay, what is happening here? Look, you have certain cells here, okay, that are either infected or they are transformed, meaning tumor cells, cancer cells. Yeah, some of you took some biology of cancer, so you should have seen some antibodies that can bind to some epitopes on cancer cells. Yeah, does that ring a bell to those of you who are taking the class?
He hasn't got that yet.
No?
No.
Oh, okay, but he will.
Yeah.
All right, and then you can sound really smart saying that you already know that stuff. Okay, how many people are taking that class? Oh, cool. Ha. All right, so here we go. What's going to happen here? On the surface of these cells, this is a nucleated cell, you have those MHC1 molecules that present a piece of protein that got processed in the cytoplasm, right? If it is self and fine done the piece of protein, you're not going to activate anything. But if it is a piece of protein from a virus, now you're going to show this piece of protein on the surface, okay? Okay, sorry, I'm mixing up the terms here of the cell killing. All right. MHC is only for the T cell presentation, right? Here you have to have the whole protein expressed on the surface. Okay, don't get those lines crossed. Easy to get confused. You have to have the whole process, the whole protein placed on the surface here. So let's say the virus is getting ready to bud out of the cell. Okay, in order for the virus to bud out of the cell, it is going to express proteins. It's going to put it on the surface, and you're going to have a coat of the virus. You're going to have this encapsulation of the virus. But the protein is on the surface. The virus is going to be ready to be budding out. When the full protein is on the surface, then the antibody can recognize it, okay? Because it's not your self protein, it's some virus, or it's a piece of cancer, weird protein. When the antibody is bound, the constant region of the antibody can interact with the receptor that is present on natural killer cells. And what is the job of natural killer cells? Is to kill. That was, yeah, easy. You don't think too much. Job of natural killer cells is to get there and kill. Okay? How is it going to kill? If it binds to an antibody that recognizes a strange protein, this cell is going to put toxic stuff on the cytoplasm of that protein in order to kill it. So this is what you call antibody-mediated or antibody-dependent cell-mediated cytotoxicity. This natural killer cell is going to be cytotoxic and is going to kill that cell that has weird stuff in it, on it. Okay? So this is one effector function of antibody, ADCC. Okay, we're going to see more on this later. Okay, on how exactly they do that. Okay, very, very important function of the antibody. Okay, and here is another function here, which is degranulation. Okay, so what is the antibody doing here? So now antibodies bind to surface of big pathogens like helminths, worms. Okay. It doesn't make sense for the antibodies here to attract phagocytes because the worm is bigger than the cell. So what these antibodies do, this specific class of antibodies, it's by the way IgE, what they do, they bind to receptor on surface of cells that are specific to get there and dump toxic stuff to kill the worm. Okay, to kill the pathogen. So we are talking about eosinophils and also mast cells. Okay, so eosinophils, basophils, and those two are going to come there, especially eosinophils. And once they bind to the antibody that is tagging your pathogen, you're going to do this degranulation. Okay, so the antibody is mediating the degranulation. Okay, so this is just one big picture slide. It's a lot of information here. okay? But this gave you all these effector functions for your antibodies. And then you finally have some points here. Okay, so the question is, let's still move this thing around. Okay, which of the following antibody effector functions results in the formation of the membrane attack complex? So you have to think, what is doing the membrane attack complex? Does anyone need more time? Everybody got really quiet. You can consult your classmates just in case. I have 2 people giving me the wrong answer. You wouldn't know. Okay, so 1 person. No, everybody give me the right answer now. Anybody else need time? No, you're good? Okay, what is your answer? D. Okay. All right. And just for your practice, you can do this. I'm not going to quiz you now because that's going to take, I don't know, 10 minutes or so to get this stuff done. But practice that at home. What was that? Okay. All right. So more here details on the structure of the antibodies. Okay. You have this thing that is Y-shaped. This is kind of a primitive drawing of it. And you have 4 peptides that are going to make 1 monomer of the antibody. You have 2 peptides that are long and 2 peptides that are short. The long one you call them heavy chains, and the short ones you call them light chains. That's pretty simple. 2 heavy chains, 2 light chains. The whole protein itself is kind of bulky. Okay, so you see here like about 150 kilodaltons. That's a big protein. Okay, so this thing is kept together by covalent interactions via disulfide bonds. Okay, so you look at the heavy chains, those are going to have a disulfide bond that is going to connect the 2 heavy chains. And then you look here at the light chains. One light chain is going to be connected to a heavy chain also by disulfide bond. And same thing, so they have disulfide bonds that are connecting light and heavy, and then disulfide bonds that is connecting the heavy chain. Now, how do you get a disulfide bond?
Secondary structure.
This is part of secondary structure, stabilizing. Actually, it's more like tertiary structure. Yeah. Oxidative agents. I'm thinking about the amino acid that you need to make. Cysteine, right? Yeah, you have cysteines that make disulfide bonds. Okay. So that disulfide bond is going to give stability to, in this case, quaternary structure. Because here you're putting 2 polypeptides, stabilizing 2 polypeptides together. Okay. If it was only like you have in the next slide, or in the following one, if it is a disulfide bond that is stabilizing within a chain, then that thing stabilizes the tertiary structure. But those disulfide bonds are stabilizing quaternary structure. Okay? So important things about antibodies. They have, each of the chains has a variable region and at least 1 constant region. So the variable region is the part that is going to recognize the antigen. Okay? And how is this going to happen? You have a heavy chain and a light chain that get together, get those 2 variable regions close together, and the interaction here of those 2 variable regions is going to form 1 binding domain for antigen. So antigen is going to get there and it's going to bind to a domain that is between those 2 variable regions, one from the light, one from the heavy. Okay? So you see with this Y-shaped protein, each antibody has 2 binding sites for antigens. Okay? Each one of those binding sites here, like binding site here and binding site there, these are identical to those. Okay, so the binding site here is the same as the binding site over there. So one antibody is very specific for one type of epitope. Okay, for one type of epitope on the antigen. Right? So this is your variable region that makes the binding sites. And then when you go down here on your chain, you're going to have the constant regions. So the constant regions, not the constant region of your light chain, but the constant regions here of your heavy chain, those are going to dictate what type of antibody you're dealing with. So the constant regions of your heavy chains are going to say, well, my antibody is going to fight infections on the blood. Oh no, the other antibody is going to fight the worms. Okay? So the constant region is going to tell what type of effector function your antibody is going to have. Okay? So the constant region is going to determine what kind of antibody you have. Okay? All right, so let's do some more fragmenting here of your antibodies. If you cut the constant region of your heavy chain, if you cut the constant regions out, and you can do that by using some enzymes, okay? If you cut that out, then you end up with 2 pieces of antibodies. Okay, you end up here with your fragment that is called FAB for fragment that binds, fragment of the antibody that's going to bind stuff, and the other part here you call the F fragment for constant region. Okay, so this is your FC region, and this is your FAB for antibody binding region. Okay, so the top part is binding, the bottom part is your constant region. Okay. Now, one thing about the constant region is that when you run an experiment with it, you can actually precipitate it and crystallize it, you know, but that's like information you really don't need, so don't worry about that. So you have binding region, you have constant region. Okay, so let's look more here at your constant region of your light chains. Okay, so light chains, you're going to have variable region and constant region. Right? The constant region of your light chain, the constant region is determined by the DNA and is not going to be changed. Okay, the only region that you change to make specific antibodies is really the variable region. So the constant regions, that's it. You have whatever the genes are telling you that you should have, okay, without mutation or anything else. Okay. Now, there are 2 genes that code for the constant region of the light chain. One gene encodes for the kappa, another gene encodes for the lambda. Okay, so when you're making antibodies, you can either have kappa expression or lambda expression, okay, for your antibodies. Now, one antibody molecule is going to have both of those constant regions to be from the same gene. Okay, so you're going to have, if you have this antibody here, you're going to have either both of them kappa or you're going to have both of them lambda. Okay, but you are not going to have a kappa lambda in the same antibody. Okay, that's not going to happen. And later we're going to see the mechanism of why that is the case. Okay, so I answered that question over there. All right, so what are these genes and what kind of antibodies classes are those genes producing? Okay, so let's see here. The genes that are going to produce the different classes of antibodies are really, encoded in the DNA. So you have these genes that are given Greek names, like Greek letters. So you have, mu, delta, gamma, alpha, epsilon, Well, one good thing about that is that they are producing the types of antibodies that are going to be either M, D, G, A, or E. Okay, so you just, if you're talking about the Greek version, that's the gene. If you're talking about the Arab version or the normal letter version, then you're talking about the antibody that is produced. Okay, so this chain here is coming from the mu. You're going to have antibody that is type IgM. If this constant comes from the gamma, you're going to have antibody that is, I'm sorry, for delta. I always get those two confused. If this is coming from delta, you're going to have antibody that is IgD, and so on and so forth. Okay. Now look at this. When you get to the gamma guy, you can have different types of IgGs. At least 5 different types of IgGs, but they all get put into the same bucket of IgG. Okay. When you look at the alpha, you can have 3 types of alpha, of IgAs. Okay. Then you look at the epsilon is only 1 type. Okay. So let's see here pieces of your antibodies, right? You have light and heavy. Your light chain has a variable region and 1 constant region. So you call the variable region of your light chain VL and the constant region called CL. Now when you look at the heavy chain, you have 1 variable region per chain and then several constant regions. Depending on the type of the antibody, you're going to have either 3 or 4 constant regions. Okay? So you're going to have here 3 constant regions if you have the class of antibodies that are IgG, A, or D. Okay? You're going to have 4 if you have IgM or IgE. Okay? So in this 4 here, this is going to be, this 3 here, this is going to be on this 3. If you would have 1 more, that's going to be your CH4. That would be IgM or IgE. All right, so let's see what is happening here with your domains of your antibodies, right? So when you look here at your light chain, it is folded in a certain way, and you call that folding the immunoglobulin domain, okay? And within this one chain, you also have disulfide bonds that are important for stabilizing the tertiary structure. Okay, so in this case, your tertiary structure because it's interchain, intrachain. Okay, so you have intrachain here, you have interchain there, interchain here. Okay, and then you look at your heavy chain, you also have intrachain disulfide bonds. So it's really stabilizing the whole structure with those disulfide bonds. Okay. One important feature of your heavy chain is this area right there, like right at the neck or at the armpit of your antibody. Yeah, I guess that's a good visualization. Why? Well, because that's the area that allows the binding sites to move around. So better thing is hinge region. We don't call it the armpit, we call it the hinge region. Okay? Hinge region of the antibody is flexible. It's very important to maintain that flexibility. Okay? All right. Oh, there it is. I know I have something about the hinge region. All right. Where you're going to find it? You're going to find it right after your first constant region on your heavy chain, between the first constant and the second constant region. Okay. All right, binding sites. As we mentioned here, that is only going to happen between those variable domains here. So light and heavy, variable domains, that's one binding site, same thing on the other side over there. Okay. All right, so here is what is meant by immunoglobulin domains. When you call domain, that's how the protein is folding. Okay, so the shape of the protein fold, you call it that domain. And this thing with a lot of these beta sheets and then turns on the other side. That is called immunoglobulin domain. So this is your light chain. When you look at the constant region of the light chain, it all has the same type of folding, immunoglobulin domain. You look at the variable region, the core of the protein here has this immunoglobulin domain, but now when you look at the loops that are close to the outside, you're going to have these loops that are going to be different for every antibody. Okay, so specifically you have 3 loops that they will be mutated a lot. Those are called CDR1, CDR2, and CDR3. Okay, so these CDRs for complementarity, oh, I'll say that 5 times quickly, determining regions. Okay, CDRs. Okay, those are the areas of your variable site that are going to be modified to recognize antigens. Okay, so you have those CDRs at your light chain, and you also have CDRs at your heavy chain. And then you put those 2 together, you're going to have a unique binding site. Okay, so CDRs are here, 1, 2, 3. CDRs are there, 1, 2, 3. You have on both chains. Okay, and both of them get modified. All right. So type here of antibodies, how you find them. You can find them as a lonely thing floating around, so you can have monomeric antibodies. So which ones are monomeric? IgG, IgD, and IgE. On the surface, okay, I'll take only 2 more minutes here and then we break. On the surface of a cell, of a B cell, you're going to find IgD, and you're also going to find IgM. So IgM, when it's on the surface of the B cell, it's also monomeric. Okay? Another monomeric antibody that you can have, if you look at the blood circulation, you can have IgA as a monomer, right? So those types. And then you look at this, 1, 2, 3, 4, 5. All 5 types of antibodies have some lifetime process that they are still monomeric. Then you have the ones that are polymers. Only 2 of them to remember. You either have IgA that is secreted at mucous membranes, or you have IgM. Okay, so IgA, when it's in the blood, is monomer. When it gets secreted at mucous sites, it's a dimer. Okay, IgM, floating in the blood, is a pentamer, and it gets secreted also as a pentamer. Okay, so these guys here have 5 antibody molecules that join. They join and they have one joining chain right there. And these guys here, they also join with the joining chain. Okay, and the presence of this joining chain is what is important to allow secretion of the antibody across mucous membranes. Okay, so either you have monomer, dimer, or pentamer. All right, so I'm going to stop here. We'll pick up from this slide.